Moving to the next question. Here the question is, the term used for transfer of pollen grains from anther of one plant to stigma of a different plant, which during pollination brings genetically different types of pollen grains to stigma, is option number 1 geitonogamy, option number 2 chasmogamy, option number 3 cleistogamy, and option number 4 genogamy. So student, here which option is the correct one? Now student, here we know that correct answer, it is option number 4 genogamy. Why we can say option number 4 is the correct one? Because the answer comes from the definition of the genogamy. So let us describe the term genogamy. We know that student, it is the transfer of pollen grains. It is the transfer of pollen grains from anther of one plant to the stigma of a different plant. It brings genetically different types of pollens, different types of pollen grains to stigma. We know that student, if we describe the term genogamy, we know that it is the transfer of pollen grain from the anther of one plant to the stigma of different plant, and it brings genetically different types of pollen grains on the stigma. So student, here correct answer, it is option number 4 genogamy.
Plants follow different pathway in the response to the environments and phase of life to form different kind of structure. Response, environment of phases of life from different kind of structure. This ability is called plasticity related to plant physiology equations. Means correct answer is plasticity.
Now student, let us come to the next question. Here the question is, which of the following algae contains mannitol as reserve food material? Here the options are option number 1 Gracilaria, option number 2 Volvox, option number 3 Eulothrix, and option number 4 Ectocarpus. So student, here which option is the correct one? Now student, here we are talking the algae which store the food as mannitol. We know that student, it is brown algae which is called Phaeophyceae. Phaeophyceae is the brown algae which store the food as mannitol and laminarin. So student, in Phaeophyceae, that means in brown algae, the food is stored in the form of the mannitol and in laminarin. So, and we know that example of the brown algae. Here, if we are talking about the Gracilaria, so Gracilaria is red algae, which is Rhodophyceae. Volvox is green algae, which is called Eulothrix. It is also called green algae. We know that student, red algae is called Rhodophyceae, whereas green algae, it is called Chlorophyceae. Here Ectocarpus is the brown algae. Ectocarpus is the brown algae, which is also called Phaeophyceae. And we know that, student, in brown algae, in Phaeophyceae, the food is stored, it is stored in form of mannitol and laminarin. So here, correct answer, it is option number 4, Ectocarpus.
Now over to the next question, and that is complete the flowchart on central dogma. Now central dogma, where we see in a particular cell, including your nucleus as well as cytoplasm, because both are involving in the complete procedure of central dogma. So here we are, the flowchart is from DNA to DNA. We have to find out when from DNA to DNA formation, that procedure or process is called replication. And from formation of mRNA from DNA, that is called transcription. Formation of D from mRNA, we know that mRNA translate into protein, and that process is called translation. So this will be protein, this will be your translation, and B, this is transcription, and this is your replication. So following the above information what I have written down over here, let's follow the options. First option is A, translation, B, replication, C, transcription, D, transduction. You can see this is wrong given over here. Second option, A, replication, yes. B, transcription, that is also correct. C, translation, yes. D, protein. So the correct answer we get over here in the option number 2. But before that, just follow the other options also. Third, A, transduction, B, translation, C, replication, and D, protein, incorrect. Fourth, A, replication, B, transcription, C, transduction, and D, protein, incorrect. So the correct answer for this question is option number 2. Thank you.
Now student, let us move to the next question. Here the question is, which of the following plants is monoecious? Here the options are option number 1, Cara, option number 2, Markensia polymorpha, option number 3, Cycas circinalis, and option number 4, Carica papaya. So student, which option here is the correct one? Now student, if you are talking about the term monoecious, so what does the term monoecious means? So let us see student, we know that in some flowering plants, both male and female flowers are present on same individual. It is called monoecious. We know that student, in some flowering plants, both male and female flowers are present on the same individual, then it is called monoecious. An example of the monoecious, it is Cara, which is monoecious plant, because the plant of the Cara having both male and female flowers on the same individual. So student, here correct answer, it is option number 1, Cara. That is option number 1, correct answer. Now student, let us come to the next question. Here the question is, mutation in plant cells can be induced by. Here the options are option number 1, infrared rays, option number 2, gamma rays, option number 3, G18, and option number 4, kinetin. So student, which option here is the correct one? We know that student, the gamma rays can induce the gamma rays. It is the gamma rays which can induce mutation, in plant cell. So student, it is the gamma rays which can induce the mutation in the plant cell. So here it is correct answer, it is option number 2, gamma rays.
Over to the next question, that is, which of the following is a correct sequence of steps in a PCR, that is polymerase chain reaction. So in polymerase chain reaction, what we do, we amplify the genetic material, that is DNA. Okay, now let's see, we have to find out the options or the steps which are performed in PCR. Let's see. First, denaturation, extension and annealing, incorrect. Second, extension, denaturation, then annealing, it is also incorrect. Third, annealing, then denaturation and after that extension, this is incorrect. Fourth, denaturation, annealing, extension, yes it is. So the fourth option is the correct answer for this question, because PCR is working on the principle of replication, and you all know that what is the steps of replication, and same are the steps of your PCR. So first step is the helical form of DNA. First it get denaturate or unwind. So first step will be denaturation. After that, what will happen, the primer will attach over here, and the attaching of this primer, that is called annealing part. After that, when a primer is attached over here, the third step which will follow, the extension of that particular primer or the addition of new nucleotides to form new copy or the carbon copy of the previous DNA. So this is how we are amplifying the DNA, and the steps are denaturation followed by annealing followed by your extension. So that's why the correct answer for this question is your fourth option. Thank you.
Now student, let us come to the next question. Here the question is, genera like Selaginella and Salvinia produce two kinds of spores. Such plants are known as. Here the options are option number 1 Heterosaurus, option number 2 Homosporus, option number 3 Heterosporus, and option number 4 Homosaurus. So student, here which option is the correct one? We know that if we are talking about the genera like Selaginella and Salvinia, we know that both are the plants are pteridophytes. Both are pteridophyte plant. Pteridophytes. Example, here it is mentioned Selaginella and Salvinia. We know that student, if we are talking about both of this plant which comes from the plant pteridophyte, that is Selaginella and Salvinia, both are heterosporous. Selaginella and Salvinia are heterosporous. It means it produces two kinds of spores. It produces two kinds of spore. It produces two kinds of spores. One is macro, which is the large spores, and other one is micro, which is being a small spores. So the production of two different types of the spores, it is called heterosporous, and it is seen in the pteridophytes like the Selaginella, like the Salvinia. So student, here correct answer, it is option number 3 heterosporous. Now student, let us come to the next question. Here the question is, which of the following stages of meiosis involves division of centromere? Here the options are option number 1 metaphase 2, option number 2 anaphase 2, option number 3 telophase 2, and option number 4 metaphase 1. So student, here which option is the correct one? Now student, if we are talking about the meiosis, we know that student, in meiosis there will be meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. And we know that student, if we are talking about the division of the centromere, we know that division of the centromere occur in the anaphase. If we are talking about the division of centromere, division of centromere. It should occur in anaphase. Here in meiosis 1 there is anaphase 1, and in meiosis 2 here is anaphase 2. Anaphase 2. Remember student, in anaphase 1 division of centromere doesn't occur. Only the segregation of the homologous chromosome should occur in the anaphase 1. But in anaphase 2 of meiosis 2, division of centromere occur. Division of centromere occur. Division of centromere occur due to pushing and pulling of the chromosomes. So in anaphase 2, the division of the centromere should occur, or splitting of the centromere should occur due to the pushing and pulling of the chromosome, but it is absent in the anaphase 1. So student, here correct answer, it is option number 2, anaphase 2.
Over to our next question, that is, during the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out. Now, don't get confused in recombinant DNA technology, because some of you will confused in that particular part, recombinant DNA technology. Actually, the question is very straight. They are asking you, for purification of your DNA, the chilled ethanol is used to precipitate out what? So let's see the options. First option DNA, second histones, third polysaccharide, and fourth is RNA. So the correct answer for this question is your first option, that is the DNA, because the recombinant DNA technology, the first step, you have to isolate the DNA which you want to recombine. Okay, so isolation of DNA is the first part of this, and in isolation of DNA, what we are taking when we perform centrifugation and various steps to find out or to isolate the DNA, the last step is to take out or precipitate out the DNA from the particular supernatant, okay, which you are having in the test tube. So when we pour chilled ethanol inside it, what happens? The DNA, it precipitate out. Rather, all histone, polysaccharide, RNA, they removed by the centrifugation process. So chilled ethanol is used for the isolation or purification of DNA. So that's why the correct answer for this question is your option number 1, that is DNA. Thank you. Now over to our next question, that is the factor that leads to founder effect in a population is. So before looking to the options, just we should know what is actually founder effect. So founder effect is the loss of variation, loss of genetic variation, we can call it, loss of genetic variation in a large population. So this is a large population. So loss of genetic variation in a large population by the entry of a small population. This is your founder effect. Now moving on to the options, because question is asking that what is the factor that leads to founder effect, what is depend or the change in the or the loss of genetic variation, that depends on which factor they are talking about. So first is genetic recombination, second mutation, third genetic drift, and fourth is natural selection. So the correct answer for this question is the third option, that is genetic drift. Why is it so? Because the entry of the small population into the large population, there is a loss of genetic variation, because the small population is more sensitive towards the increase in the genetic drift. So that's why we are taking genetic drift as the correct answer for this question. While genetic recombination is not the factor for founder effect, mutation is the sudden change in a particular nucleotide. We can't say that mutation always occur in a population altogether. And the last, natural selection, that is not leading to founder effect. So the correct factor or the correct answer for this question is option number 3, that is genetic drift. Thank you.
Now student, let us come to the next question. Here the question is, which of the following algae produce carrageen? Here the options are option number 1 brown algae, option number 2 red algae, option number 3 blue green algae, and option number 4 green algae. So student, here which option is the correct one? We know that student carrageen, if we are talking about the carrageen, we know that carrageen is produced or obtained from red algae. Student, we know that carrageen is produced from the red algae, which is also called rhodophyceae. So student, here correct answer, it is option number 2 red algae. Now student, let us come to the next question. Here the question is, gamma are present in option number 1 pteridophytes, option number 2 some gymnosperms, option number 3 some liverworts, and option number 4 mosses. So student, here which option is the correct one? We know that student, if we are talking about the term gamma, we know that gamma, what type of the term it is called? Gamma. We know that student, gamma are green multicellular asexual bud which develop in a small receptacle called gamma cups located on thalli. Example, it is seen in liverworts. So student, we know that gamma are the green multicellular asexual buds which develop in small receptacles called gamma cup located on thalli. So here correct answer student, it is liverworts. Here correct answer option number 3. Now student, let us come to the next question. Here the question is, the amount of nutrients such as carbon, nitrogen, phosphorus and calcium present in the soil at any given time is referred as. Here the options are option number 1 climax community, option number 2 standing state, option number 3 standing crop, and option number 4 climax. So student, here which option is the correct one? We know that student, correct answer, it is option number 2 standing state. Why we can say option number 2 is the correct answer? Because the answer comes from the definition of the standing state. So if we describe the term standing state, we get our answer standing state. So how we can define this term? It is the amount of nutrient, it is the amount of nutrient such as carbon, nitrogen, phosphorus, such as carbon, nitrogen, phosphorus and calcium, present in the soil at any given time is called standing state. So student, here it is the amount of the nutrients such as the carbon, nitrogen, phosphorus and calcium present in the soil at any given time. It will represent the standing state. So here correct answer, it is option 2. Now student, let us come to the next question. Here the question is, when the centromere is situated in the middle of two equal arms of chromosomes, the chromosome is referred as. Here the options are option number 1 telocentric, option number 2 some metacentric, option number 3 acrocentric, and option number 4 metacentric. So student, here which option is the correct one? We know that student, if we are talking about when the centromere is situated in the middle of the two equal arms of chromosomes. So here it is the chromosomes. We know that student, when centromere is present in the centromere, when centromere is present in the center of the chromosome, so that both of the arms get equal. So such type of the chromosome, it is called metacentric chromosomes. So what will happen in metacentric chromosome, student? Metacentric chromosome. Metacentric chromosome. The centromere is at center. The centromere is at the center. Is at the center of the chromosome. So that both of the arms, get equal. So student, metacentric chromosome are those chromosomes in which centromere is situated at the center of the chromosome, so that both of the arms get equal in this side. Such type of the chromosome, it is called metacentric chromosome. Here the correct answer, it is option number 4.